Hi, Assalamualaikum. My name is Alia Maisara binti Ramzan. My matric number is S56068. Today, we are going to discuss about the topic which is diversity of marine phototroph. These are the important terms that we need to know in this topic. The first one is primary producer. Primary producer is the organism that can synthesize organic compound from inorganic substances. Phototroph. Phototroph is the organism that can grow using light as its energy source. It also can be defined as organism that can synthesize ATP and using light as its energy source. Photosynthesis is basically the process by which the plant uses sunlight, water, and carbon dioxide to produce oxygen and energy. The last one is photosynthetic organism. Photosynthetic organism is a part of phototroph, which is it can perform photosynthesis. Okay, now let's go through the chronological order of the diversity of the marine phototroph. Okay, this is the traditional and new understanding in primary producer. In mid-1970s, diatoms and flagellates were understood as a primary producer in the environment. They are the major organisms that can be collected by a phytoplankton net. Both microalgae are distributed in a wide range of water quality, including eutrophic and oligotrophic environment. What is eutrophic and oligotrophic? Eutrophic is the lake which has high nutrient and high plant growth, while oligotrophic is the lake that have low nutrient concentration and low plant growth. Why diatoms and flagellates are considered as a primary producer? This is because these two microalgae were easily speculated from the approach of phytoplankton sampling and the method of phytoplankton detection at the time. In 1970s, phytoplankton net is used to trap plankton. Basically, phytoplankton net is a conical polyethylene net with a defined mesh size and has a collecting bottle that attached to the tip end. The mesh size of the phytoplankton net determines the size range of the phytoplankton. Only the size more than 60 micrometer were used in phytoplankton research in 1970s. However, in recent phytoplankton research, deoxyribonucleic acid DNA and flow cytometry are used. Cytometry is the measurement of characteristic of the cell. As for the result, it showed that predominant phytoplankton is much smaller than the phytoplankton that had been collected by phytoplankton net. For example, hoptophytes including coccolithophores. Plankton net with a smaller mesh size around 10 micrometer had been employed for the research phytoplankton in recent years. Okay, next is the recognition of Picocyanobacteria dominans. In the late 1970s, based on the development of observation technique by fluorescent microscopy and the accumulation from electron microscopy, observation reveals that unicellular cyanobacteria was a primary producer. The unicellular cyanobacteria is Cyanococcus. It is a picocyanobacteria as it have very tiny cell. The size of this picocyanobacteria is 0.5 to 0.2 micrometer length and 0.5 to 1 micrometer diameter. In the late 1980s, development of the high-performance liquid chromatography (HLPC) technique for pigment analysis and of the cell detection technique that using cytometry technique reveals that picocyanobacteria or Cyanococcus contain divinyl chlorophyll A, which had slightly different with the absorption of spectrum from the usual chlorophyll A. Those cells did not contain picobilipigments, which is the light harvesting antenna in common Cyanococcus. And then they come up with the cyanobacteria named Prochlorococcus, and it become dominant phytoplankton in tropical and subtropical region. Prochlorococcus bacteria perform oxygenic photosynthesis using divinyl chlorophyll A or B, which can absorb the blue light. Prochlorococcus plays a significant role as a primary producer in the marine environment, as the amount of divinyl chlorophyll A is 3 times higher than chlorophyll A. Okay, let's take a look to the discovery of ubiquitous photoheterotroph. In early 2000s, there are 2 significant findings that prove that microorganisms can transform light energy into chemical energy. The first one is the distribution of aerobic anoxygenic photosynthetic bacteria, AAPB, and another one is the distribution of proteohedopsin containing bacteria. Both bacteria are photoheterotroph without carbon fixation and had never been recognized as the primary producer. What is photoheterotroph? Photoheterotroph is basically a combination of phototroph and heterotroph. It can be defined as organisms that gain their energy from the sunlight and getting their carbon from organic sources in the environment. AAPB was derived from non-sulfur purple bacteria. AAPB also contribute to the transformation of solar energy and act as primary producer. And aerobic photosynthesis is the process by which certain bacteria use light energy to create organic compounds, but they do not produce oxygen. Proteohedopsin is the rhodopsin-like protein detected in marine proteobacteria. Rhodopsin is a photoreceptor in vertebrates. As a light sensor, it contributes adenosine triphosphate ATP synthesis. Proteohedopsin was play a significant role in light energy transformation in the marine environment. Okay, next let's move to the oxygenic photosynthesis using far red light. In 1990s, chlorophyll A is essential for oxygenic photosynthesis, such as light harvesting, energy transfer, and charge separation in the primary reaction of the photosynthesis. Chlorophyll A can estimate the amount of oxygenic phototroph and the activity of oxygenic photosynthesis. Photosynthetically active radiation, PAR, evaluate that the light strength for the oxygenic photosynthesis. In 1996, chlorophyll D was exist. There was a cyanobacteria, which is Acariochloris marina, that use chlorophyll D to perform oxygenic photosynthesis. This cyanobacteria is a oxygenic photosynthetic bacteria that contain light harvesting pigment and absorb carbon dioxide and produce oxygen. Chlorophyll D is a redshift chlorophyll that can absorb far red light for an almost 30 nanometer wavelength, longer than chlorophyll A. Photosynthesis that using far red light contribute to the primary production for the marine environment. In 2010, there was another redshift chlorophyll, which is chlorophyll F. Chlorophyll F was reported from a cyanobacterium. It can absorb far red light for an almost 760 nanometer. The discovery of eukaryotic phytoplankton. In recent years, very diverse of eukaryotic phytoplankton species have been revealed. Only some dozen of species in Chlorophyta, Heteroconophyta, and Haptophyta were known as eukaryotic phytoplankton until the 2000s. Around 2010, several reports on marine phytoplankton analysis using the technique of metagenomes and flow cytometry were published. Metagenome is a direct genetic analysis of genomes contained with an environmental sample. This shows that a significant amount of phytophytes, including many undescribed species, were widely distributed in marine environments. In 2007, phytoplankton which did not belong to any known phyla of algae were discovered by metagenomic analysis of a plankton fraction. The phycobelophytes distribute widely in the coastal area of Europe and North Atlantic. In 2011, new candidate phylum was also detected by metagenomic analysis that targeting the plastic DNA, which is Rhapomonads. Rhapomonads were close to the haptophytes, but they did not belong to any known phyla phylogenetically. Strange phototrophic microorganisms. The cyanobacterium-like particle, Eucin A, was first detected by metagenomic analysis of nitrogen fixings bacteria in marine waters. Eucin A indicates that the particle contributes to the nitrogen cycle in pelagic water. Usual cyanobacteria grow by oxygenic photosynthesis process photosystem, a respiration system, and genes for a carbon fixation system. Rubisco is the primary enzyme for fixing CO2 and the 3-carboxylic acid cycle for respiration. While Eucin A is a strange phototropic microorganism. It is because Eucin A might be a symbiont in some organisms due to the lacking of photosystem 2. But since it possesses a complete gene set for photosystem 1 and contains chlorophyll, it must use light energy for some activity of the particle.